Hello and welcome to Taconic's webinar titled The Influence of the Microbiota on Immune Response, which will be presented by Dr. Ivanov from Columbia University. My name is Jennifer Phelan and I will be your moderator today. Before we get started, I'd like to address a few practical matters. This webinar will be made available to all attendees. We will email a link to the presentation to you by the end of this week. Directly after the presentation, Dr. Ivanov will answer your questions as they appear in the control panel. So if you have a question, please type it in the control panel. The other attendees will not be able to see your question as it is sent directly to Dr. Ivanov and I. If you cannot see the control panel, just single-click on the single arrow on the top of your screen. In case your question is not answered at the end of today's session, we will respond to you within the next 48 hours via email. If you are experiencing technical difficulties, please send the host a chat message or raise your hand and our staff will be able to assist you. We hope you will enjoy today's webinar about the influence of the microbiota on immune response. So let's get started.
Okay, hello everybody. My name is Ivailo Ivanov and I'm an assistant professor at the Department of Microbiology and Immunology at Columbia University. And let me see if I can, yes. So most of the talk we will talk about a specific example that we identified of how this particular commensal bacteria affects immune responses and how and maybe we'll have a discussion of how microbiota may affect your experiments and what we should do about it. But I think before we go into that, I think it's important to point out that the interaction between microbes and the organism are really fundamental and really important and crucial for life as we know it. And in fact, most multicellular organisms, starting from plants and insects, all the way up to mammals and human, of course, have learned to depend on microbes for their biological function. So we not only share a table with these commensal bacteria, but we also depend on their presence for our biological functions. And when I think about the three main features of what we can think of commensal bacteria that are important to kind of keep in mind is the first that there are a lot of them. So there are very high numbers of microbes living on our bodies and in our mice, of course. And the second feature is that what we will be talking about is resident bacteria. So, of course, we get exposed to microbes on a regular basis, most of which are transient. But what we're talking here about is bacteria that have evolved with the host, that are intimately associated with host tissues. As you can see on this electron micrograph, which you can see the mucus layer on top of the epithelial cells in the colon, and you can see inside this mucus layer there is a community of bacteria, and now we know also of viruses and fungi that are really intricately and intimately associated with the host tissue, and some people refer to them as microbial organ. And the third feature that is important is the fact that this community is non-random, and in fact the composition of this community is important, as we will see, and that may affect biological function of the host. Most of these organisms are species that have evolved to colonize this particular niche of the environment, which is our intestine or skin or other mucosal organs. But also they have been selected by the host, or we think they have co-evolved with the host based on the functions they may provide. And just to illustrate, of course, we now know that there are so many bacteria, but just to illustrate that, we know that, for example, in the gastrointestinal tract, we have the gastrointestinal tract is designed to interact with the outside environment, and there are anatomical features that allow for the increase of the surface of this interaction. And we know that, for example, in human, the intestinal area is around 300 square meters, which roughly translates to 50,000 petri dishes filled with bacteria. So you can think about the enormous amount of microbes living in this environment. And in fact, we know that the number of microbial cells, in fact, outnumbers the number of eukaryotic cells by an order of magnitude. But even more importantly, the number of genes that may encode important functions that bacteria, that this microbial community encodes, outnumbers the number of human genes by 2 orders of magnitude. In terms of immune functions, we know that commensal bacteria are important for providing protection against pathogens, against infections, and this can be generally summarized in 3 ways. First, we know that commensals, by the sheer numbers of them, may occupy niches for pathogens and thus prevent pathogens from establishing an infection. Certain bacteria may produce substances, commensal bacteria may produce substances that directly inhibit the growth of a pathogen, such as changing the pH, for example, or other bacteriostatic substances. And the third most important way by which commensals provide immune functions is that they can stimulate the immune defenses of the host, or they can affect the immune repertoire of the hosts. And I have listed here several ways by which we know they can do that. They are important for establishing of the normal anatomy of the immune system in the mucosa. They can stimulate epithelial cells to produce antimicrobial peptides and mucus, which are important in the defense against pathogenic bacteria. They can also stimulate the immune system to secrete molecules such as secretory IgA, or they can induce recruitment of lymphoid cells to the gut or the mucosa. In addition, we now know that they may have systemic effects, such as affecting the balance of systemic helper T cells and their function. So what we have been interested in my laboratory is this kind of interaction between these 3 types of players. We have the commensal bacteria, or the resident microbial community, we have pathogens, and you have the immune cells. And these 3 players are, and these interactions occur across a single layer of epithelial cells that divides the enormous amount of microbes and other antigens from the tissue or the immune system, which is present in the lamina propria of the intestine. And there are feedback mechanisms and interaction between all these players, but what we have been interested in is how the composition of commensal bacteria may affect the immune composition of the immune system, or important in the way the organism responds to infection or to another immunological challenge, such as autoimmune inflammation or other pathogenic conditions. So we've been focusing on the system of T cell homeostasis in the gut, and we know that CD4 T cells develop to different effector fates under the control of different environmental conditions, such as different cytokines produced by innate immune cells. And we recognize several types of effector T cells that, depending on their function, we know the Th1 cells are responsible for killing intracellular pathogens, Th2 cells respond to helminths, and the two cell types that we've been focused on are Th17 cells, which are important for clearing extracellular pathogens such as bacteria, especially fungi at mucosal surfaces, and regulatory T cells, which are of course important for suppressing or tuning down effector responses once the infection is cleared. Of course, dysregulation of these two cell types can lead to disease and uncontrollable expansion of Th17 cells, leads to autoimmune inflammation, and of course, lack of regulatory T cells will lead to also to autoimmunity. It's important to kind of keep in mind that these two cell types share not only common developmental pathway, but they share common separation of function. There's an yin and yang, a balance between the two. These two cell types establishes the type of immune response or the type of immunity that is generated at any particular time. And this is even intricately woven into the interaction between the two major transcription factors. In fact, it has been shown that they are expressed in precursors at the same stage, in the same cell, and interact and inhibit each other. And depending on the cytokine environment, one of them can prevail and lead to either Th17 and regulatory T cells. So there's balance between these two cell types. And we've been interested in Th17 cells because they're important in a number of autoimmune inflammatory conditions. This is just a short list of conditions in which Th17 cells have been shown to play a pathogenic role and associated with or function of Th17 cells in immunity. It is considered that they provide protection against mucosal infections, and this is mainly mediated by the cytokines that they produce. The two major cytokines are IL-17 and IL-22. IL-17 is important for basically recruiting neutrophils and jump-starting inflammation. And IL-22 acts mainly on epithelial cells, which express the IL-22 receptor and react through the response to the production of antimicrobial peptides and other effector cytokines, which increase mucosal protection. However, it should be pointed out that the exact role of Th17 cells in all of these conditions is not completely clear because, of course, the effector cytokines, both IL-17 and IL-22, have now been shown to be produced by other cell types in addition to Th17 cells. What was interesting to us was the fact that both regulatory T cells and Th17 cells are highly enriched in the gut at steady state. And of course, we know that regulatory T cells are important in the intestinal environment. They're important suppressors and controllers of inflammation, simply because of the fact that there is such high antigenic load, and under steady state they need to control immune responses. And this just shows you that regulatory T cells, which are labeled here by the expression of FOXP3, a transcription factor FOXP3, are present in all tissues. However, they are enriched in the small intestine, especially in the colon, where 40 to 60% of the CD4 T cells may be regulatory T cells. But interestingly, similarly to regulatory T cells, when we looked at the representation and distribution of Th17 cells at steady state, we found that even though Th17 cells were present, or IL-17 producing cells were present in all tissues, only in the intestinal tissues including small intestine and colon, they represented a significant proportion of the IL-17 producing T cells, CD4 T cells. So we decided to ask whether intestinal microbiota may control this homeostasis, this ratio between Th17 cells and regulatory T cells, and if that is the case, whether individual bacteria are responsible for this effect, and finally, what is the mechanism of this induction. And I'm just going to show a few slides just to show how we address that, because I think that's a good example of how the role of microbiota and identification of a particular member of the microbiota can be carried out when an effect of microbiota is suspected on your experiments. So the first experiment and the first proof of principle is to whether microbiota is important to treat the mice with antibiotics. And here we treated just regular 57 black 6 mice with a cocktail of antibiotics. This is the green A column. A is for all antibiotics. As you can see, when at steady state, without treatment, no treatment, there is a certain percentage of Th17 cells in the lamina propria. If mice are treated from birth, and we can talk about why from birth, but with the antibiotics, which prevents, in fact, the expansion of gut bacteria and brings the numbers of gut bacteria down considerably, that prevents actually the generation of Th17 cells. Another way to do this experiment is to test whether individual antibiotics may be important for the effect, and indeed when we treat mice with vancomycin, one of the antibiotics in the cocktail, we can replicate the effect, meaning we block Th17 cell differentiation. The second part of this proof of principle is to show that that this effect is not due to just systemic effects of the antibiotic. And one way to do that is what we did in the right part of this experiment, where after we treat with antibiotic, we see depletion of Th17 cells, then we stop the antibiotic treatment, and then the mice are split in two groups. One of them is just kept on regular water without antibiotics, and as you can see here, these mice do not recover Th17 cells, and another group is placed basically in a cage. In fact, there is in a cage with a couple of mice that have not been treated with antibiotics, which allows for the horizontal transfer of bacteria, or we can do that with actually doing a fecal transplant by gavaging the mice with feces from other mice that have not been treated with antibiotic. And this now recovers Th17 cells. So this strongly argues that the effect that we see on Th17 cells is indeed due to the presence of commensal bacteria. It is also important to do a second experiment, and this is experiment in autobiotic or germ-free mice. And we can discuss the reasons for that, but it has to be confirmed. And in this case, we compared conventionally raised mice to germ-free animals. And again, as you can see in the germ-free animals, Th17 cells are virtually absent from the small intestinal lamina propria. Again, because germ-free animals have multiple developmental defects, it is important to provide the proof of principle where you bring back the bacteria. In this case, we gavage germ-free animals with bacteria with feces from conventionally raised mice, and we can see a nice recovery of Th17 cells. And this proves that the microbiota at steady state is important for supporting Th17 cell development. So the next question we asked is whether this is due just to presence of bacteria, or is it the presence of specific bacteria? And the first set of experiments that we performed was to colonize germ-free mice with individual bacteria. And in fact, we performed a number of experiments in which we either colonized with individual bacteria or with combination of bacteria that we can grow in the lab. And none of these experiments yielded recovery of Th17 cells. So you can see here an example when we use Enterococcus fecalis that we cannot recover Th17 cells, in contrast to what happens when you give a full complement of commensal bacteria from conventionally raised mice. So the conclusion of this experiment is that there are two possibilities. One is that you need a very diverse community of bacteria that we cannot, of course, replicate in these individual mono or dual colonizations. Or the other, of course, alternative possibility is that there is a bacteria that we have not tried that is responsible, or a set of bacteria that is responsible for this effect. So at that point we decided to follow the second possibility first, or to test it, and in fact we tested if that is the case, if there's a certain bacteria that is mediating the effect, then one would expect that mice that do not have this bacteria will not show Th17 cells. So we in fact evaluated mice, 57 black 6 mice from different facilities. We first used a number of academic facilities in the New York area and at the NIH, and all of them had Th17 cells. We also tested certain vendors, mouse vendors, and interestingly, when we tested 57 black 6 mice from Taconic versus Jackson, we saw a very discrepant, a very differential representation of Th17 cells. Taconic B6 mice, as you can see here on this slide, have high levels of Th17 cells in the lamina propria, in contrast to Jackson B6 mice, which is the same strain of 57 black 6 you just obtained from Jackson, had low levels of Th17 cells. And we could show that this is indeed not due to a genetic drift or a genetic difference between the two types of mice by performing this particular experiment and a number of other experiments, but this is kind of the crucial experiment where we take feces from Jackson or Taconic mice and we transfer them into germ-free mice. And as you can see here, again, colonization of germ-free mice with Taconic microbiota from Taconic B6 mice, wild-type levels of Th17 cells, and in contrast, colonization with Jackson microbiota or fecal material from Jackson B6 mice leads to a very low recovery of Th17 cells. And in fact, the difference between the conventionalized, what we call these conventionalized mice, is very similar to what happens to the conventionally raised Jackson and Taconic B6 mice. So I think this is important, and we can discuss maybe if there are questions of how this may affect your experiments, but obviously you can see that depending on what controls you use, if you use Jackson B6 or Taconic B6, you may come to very different conclusions of the effects of Th17 cells in your particular model. And so these experiments, we can show two things. One is that Th17 cells at steady state in the small intestinal lamina propria are induced by signals from commensal bacteria, and the second one is that there must be a Th17 cell inducing component of the microbiota that is, by simplest explanation, is that that's bacteria that's present in Taconic B6 mice, but it's absent in Jackson B6 mice, at least in the ones that we examined at the time. So in order to identify these bacteria, we in fact simply compare the microbiota between Taconic B6 and Jackson B6 mice. And we used, I believe, 4 mice of each location, of each vendor. They are all 67 black 6 mice, and this is a representation of the differences between the two groups. In fact, it's very easy to tell where a mouse is coming from based on its microbiota because they're very different. As you can see here, what's labeled on this figure with green are species that were overrepresented in the taconic B6 mice, but were underrepresented in Jackson B6 mice, and with red is the opposite. Species or taxonomic units that were overrepresented in Jackson mice and underrepresented in taconic mice. In fact, you can see that the Jackson microbiota is somewhat more more diverse in this figure, but there are individual OTUs that are overrepresented in taconic mice, and we were really interested in bacteria, or in this case we are only looking at bacteria that are in fact present versus absent, in so very large differences between the two groups. And the top two candidates were Lactobacillus murinus and this species Candidatus artemitus. We had already tested Lactobacillus murinus in our monocolonization experiment, so we could eliminate that as a possibility. And so we focused on this Candidatus artemitus, or segmented filamentous bacteria. And what are the segmented filamentous bacteria? I will call them SFB from now on. They're an interesting type of commensal, or member of the resident microbiota in a number of species. Oops, I think I'm missing. I apologize, I'm missing a slide, but they have a very unique morphology. They form these long filaments. Another interesting feature, a very specific feature of SFB, is that they interact closely with the epithelial cells. Interestingly, these bacteria have been identified a long time ago, but have remained understudied for a while because they cannot be cultured in the laboratory. They're closely related to Clostridia, to the Clostridia family. They are gram-positive anaerobes, and they have been shown to perform certain immunomodulatory functions. They have been shown to induce class II expression on epithelial cells, and also the induction of secretory IgA and recruitment of intraepithelial lymphocytes. Because of their unique morphology, we actually decided first to look whether they're present or absent, where their presence correlates with the induction of Th17 cells, and this is an electron micrograph of the terminal ileum of a Jackson B6 mouse after washing the most of the luminal contents. And this is the same section of the intestine from that we had at the time, and you can see the very different morphology and very obvious difference between the representation of bacteria that have the morphology of SFB. So this is a close-up of the interaction between SFB and the epithelial cells. Just wanted to point out that in contrast to other microbes, SFB are mucosal associated. They interact closely with the epithelial cells. However, they are non-invasive in the sense that we could never find penetration of the bacteria inside the lamina propria or even within the epithelial cell. Also, they are non-inflammatory in the sense that they don't cause any inflammation in germ-free mice by themselves. But as you can see, they will contribute to autoimmune inflammatory conditions through their effects on the immune system. Again, in order to prove that this bacteria is the one causing the effect, in our case Th17 cells, to fulfill these Koch's postulates, we needed to colonize germ-free mice with a purified culture of the bacteria. Again, as I mentioned, SFB are unculturable. At least at the time, they were not culturable. So the way we solve this problem is by collaborating with a Japanese company called Yakult, who had back in the 90s generated SFB mono-associated animals. So they had generated mice, kept them in isolators that only have SFB and no other microbe, and we have since tested that, and indeed they don't have any other bacteria in their feces except SFB. So using fecal material from these animals, we colonized germ-free mice, and we were able to show that, in contrast to all the other experiments and all the other bacterial species that we tried, SFB can by itself, as a monocolonization, induce Th17 cells. It can induce both IL-17 and IL-22 production from CD4 T cells, very similar to levels that are very similar to the steady state levels in conventionally raised mice. The ability of SFB to induce Th17 cells turned out to be specific in the sense that, first of all, it is the only known commensal species that we know at this point that can induce Th17 cells. Even closely related Clostridia do not have this effect. And at the same time, it's specific for Th17 cells because the response is mostly in the Th17 cell type, and we do not see any significant difference in the numbers of Th1 or the proportions of Tregs or Th2 cells, other CD4 effector T cells. So what is the biological consequence of having SFB? So we could show that mice colonized with SFB are protected from infection with intestinal pathogens. In this case, in the left side, you're looking at an experiment just taking taconic mice and Jackson B6 mice, or mice that don't have SFB and mice that have SFB, and infecting them with Citrobacter rodentium. And as you can see, the mice that colonized with SFB show lower levels of colonization of the pathogen. And this has now been shown for other, reported for other models of infection, that SFB in certain models can provide protection against certain infections. And on the right, what you can see is the fact that SFB, on the other hand, by inducing, or presumably by inducing Th17 cells, can increase the susceptibility to autoimmune inflammation. And in this case, we use the model developed by Diane Mattis and Christophe Benoit. It's a transgenic model called a KB by N or KRN model, in which we have a transgenic TCR recognizing an antigen in the joints, inducing arthritis. So these mice develop spontaneous arthritis. But interestingly, what Diane and Christophe found is that these mice do not develop arthritis when they are re-derived germ-free. And when in collaboration with them, we colonize these mice with different bacteria. Again, colonization with control feces, such as Jackson feces in this case, didn't induce disease in the germ-free transgenic animals. However, colonization with SFB leads to Th17 cell induction, IL-17 production, and development of disease within a few days of colonization. So, in summary, what I've shown you is that Th17 cells are indeed induced by signals from a specific component of the microbiota, and through these types of experiments, we can indeed identify such components. In our case, we identified SFB, and even if these components are not culturable, in this case, we still can only propagate SFB mostly in monocolonized mice. And the presence of these individual members of the microbiota, which may change immune functions or the immune homeostasis, has important consequences for not only the immunological fitness, but just in general in the response of the organism to immunological challenge, such as infection or autoimmune model, disease model. And similarly, a few years later, the group of Kenya Honda showed that regulatory T cells in the gut are controlled by a different member of the commensal microbiota. In this case, they identified certain species of Clostridia different from SFB that, as shown here, can promote the expansion of regulatory T cells in the colon. And I believe that this concept that the composition of the community on one side of the epithelial barrier, this microbial community, can affect the function or numbers or balance in the community on the other side of the epithelial balance in the sense of the immune cell community. This concept is now very well accepted, and we now know of that the composition of the microbiota controls almost every single cell type in terms of their abundance or function in the intestinal mucosa and also systemically. So just because we were talking about SFB, I just want to show you, interestingly, right now SFB is included in the health status of taconic animals, and this is just a quick search that I did the other night showing you the 57 black 6 production locations at taconic, and as you can see with green here is labeled are locations that don't have SAB, and with red locations that do have SAB. So the point I wanted to make with this slide is that the source of the animals, such as the vendor, does not necessarily equate to the presence or absence of the bacteria, in this case SFB, even though in our experiments we were ordering them from taconic. It just so happens that it was one of the rooms that has SFB, but now we know that every single room, even within a location, within even the same company or even within the same academic facility, has a very different microbiota. And in fact, even different rooms in the different racks in the same room may have different microbiota. So it's important to know that you cannot equate SAB presence with taconic mice and SAB absence with Jackson mice. Let's see. This the importance of the composition of the bacteria may be even more far-reaching. They can explain certain geographical differences in immune cell balance. For example, we know that Th17 cells are abundant in the small intestine, and regulatory T cells are abundant in the colon, and so this balance is shifted in the two geographical locations. Interestingly, the balance between the bacteria that are important for induction of these two cell types is also geographically different. SFB interacts with epithelial cells exclusively in the terminal ileum, as we show in a couple of slides, and the Clostridia that induce regulatory T cells are mostly endemic for the colon. So this is just to show you that we recently just looked at the representation of SFB. Even though SFB, as you can see in the panel A, is present throughout the gastrointestinal tract, so it's present in the proximal and distal small intestine, is present in feces. We can easily actually detect it in feces. However, if you look at the mucosa-associated fraction of SFB, it only associates with the intestinal epithelial cells in the last half of the intestine, especially in the last tenth of the intestine of the terminal ileum in mice. And another important thing is that in laboratory mice, at least the mice that we have, which are mostly probably equal to taconic B6 mice, SFB represents virtually the only bacteria that is present in the mucosa-associated fraction. This is shown in panel C, where you can see the proportion of SFB to total bacteria. In feces, it is very small, even though it's easily identifiable by qPCR. And in the ileum, lumen is also relatively low. However, if you look at the ileum mucosa in the epithelial cells, it represents 95 to 99% of the bacteria. And just to show you again, this is a demonstration to show you that SAB only binds to epithelial cells in the terminal ileum, but not in the cecum and colon, where it's also present in the luminal contents. Interestingly, this close interaction in this particular location leads to induction of Th17 cells, or the increase in the abundance of Th17 cells in that location as well. And this just shows you the representation of Th17 cells in the different parts of the intestine. You can see they're increased both in the proximal and distal ileum compared to duodenum and jejunum in SFB positive mice. So with this, I think I will end. I had a few slides, but let me see. I would go to my last slide so I can take some questions. Let me see. I guess what I just wanted to point out that we now know that microbiota is an important therapeutic target. We know that microbiota composition affects a number of biological functions, and we want to be able to manipulate this target. However, the arsenal of weapons to manipulate these targets is relatively limited. We know that we have certain arsenal of antimicrobials. We know that we can colonize people with defined bacteria or possibly with beneficial bacteria. We know that diet is a very important modulator of this community. However, all of these interventions are relatively and in many cases irreversible or difficult to control. So what we do believe in a reductionist approach is that identifying examples such as SFB of immunomodulatory bacteria or bacteria that have specific effects on the immune system and characterizing the mechanisms of this interaction and how the bacteria exert these effects will be important in actually generating more specific targets for intervention therapeutically. So I'd like to thank the people in my lab that did most of this work and some collaborators, of course, our funding, and I'll take some questions from the audience.
Thank you, Dr. Ivanov. One of the questions we received is, could you discuss other markers on the resident Tregs other than just FOXP3?
So for the regulatory T cells, other markers in the gut. So FOXP3 is of course the quintessential marker for regulatory T cells. There are certain surface phenotypic markers that can be used to identify Tregs in the gut. In the gut, CD25 is not a very good marker because we find that not all Tregs express high levels of CD25 as opposed to in the periphery, secondary lymphoid organs, for example. But interestingly, as I mentioned, most of the Tregs that are induced in the gut, they're inducible Tregs, are indeed specific for commensal bacteria. So they recognize, it's been shown recently that they recognize commensal antigens, and that may be important for their function in suppressing commensal responses. How this is induced is unclear at this point. I'm not sure if that answers the question, but.
Okay. Did you, I'm sorry, was there a gender difference between the microbiota?
So gender difference in the microbiota would be expected, and I believe that that has been looked to some degree. In our case, SFB does not seem to be differentially represented between female or male animals, nor does it induce different types of immune responses, such as different levels of Th17 cell induction, for example, in female versus male animals. So it maybe in relation to that, of course, it will be it's interesting that, of course, females are mostly responsible for transferring microbiota vertically, and they also will have more effect on the microbiota of the pups because they also deliver certain IgAs and things like that to the pups. So the microbiota of the females will have important, presumably, effects on the pups themselves. And the other thing to also consider is the fact that we can co-house females, but we don't co-house males. So, for example, when we wean pups, we sometimes may put females from different litters together, which we usually don't do with males, especially if they're adult males, or we can combine mice from different cages, females from different cages into the same cage. So that will lead to a mixture of them, or mixing of the microbiota. So it's something to consider when you suspecting effects of microbiota, that usually the bacteria, the microbiota components will transfer with the mice. So if you put females together, they will equilibrate their microbiota.
Okay, thank you. And apart from arthritis, can you list any other disease models that appear to be directly affected by the presence or absence of SFB?
Yes, I mean, as you can imagine, all models that involve Th17 cells may be affected. And the simplest example is the EAE model, which was shown to be affected by the presence of SFB. But I think just in general, another model, I think in general, all of the immune models, I think standardization of the microbiota will be very important in any immune model, in any disease model. In fact, we now do all our experiments. One way to do that is to always use littermate mice and always use co-house littermates. So basically try to equilibrate the microbiota as much as possible, because many of these models, all colitis models, are microbiota dependent. We don't know if they are specifically SFB dependent, but we know that all colitis models are microbiota dependent. And not only that they depend on the presence of bacteria, but they may depend on the presence of specific bacteria. So there is a lot of examples in which rederivation of a line in a different facility leads to change in the response during a colitis model, for example. So I think great care has to be taken when people set up this kind of experiments. The example here, just because I have a couple of minutes, is the way we discovered the Jackson versus Taconic differences is that we were comparing a knockout model that came from Jackson, and at that time we didn't co-house our animals. So our conclusion is, by comparing a knockout from Jackson to our resident wild-type mice, our conclusion was that the model lacks Th17 cells, but that was simply due to the fact that we used the wrong wild-type control that happened to have Th17 cells. So I think it's very important for methods to be developed to standardize experimental setups based on standardizing the microbiota between the different models. The same way we standardize our models in terms of genetics by using only B6 or BALB/c mice, the same genetic background. It's important to make sure that the microbial background is the same.
Okay, so then we have questions on, can you discuss the role of SFB in humans, and also are there differences in SFB between humans and mice, and can they cross-colonize?
Yes, so there are a couple of questions. So SFB, the presence of SFB in humans has been difficult to kind of ascertain, and the now there are at least 3 publications that showing that SFB is indeed present in humans. Whether it is the same type of bacteria as in mice is unclear. However, SFB is present in almost every single animal species examined to date, starting from insects, fish, birds, rodents, of course, and mammals. And interestingly, SFB seems to be species specific, so it seems to be host adapted. In fact, there are old publications showing that even SFB from rats cannot very well colonize mice and vice versa. So if SFB is indeed present in humans, it's most likely that it's adapted to this particular host, and cross-colonization may not elicit the same effects as same species, bacteria from the same species. Yes.
Okay. Is SFB thought to be probiotic?
That's also an interesting question, but difficult to unidirectionally answer. We consider SFB as having certain effects on the immune system, and in some cases these effects are beneficial, such as maybe protecting from infection, but in some cases these effects can be detrimental, such as in our making the mice more susceptible to autoimmune inflammation, which is the same for any type of immunity. Are Th1 cells beneficial or not? Th1 cells may protect you from certain infections, but of course dysregulation of this pathway will lead to disease. So I think it's similar for gut bacteria as well, and that is why it is probably not viable to think of using bacteria itself as a probiotic. And what we are trying to basically dissect is the mechanism, to use the mechanism to find molecular targets, as opposed to using the whole bug, which in some cases may be detrimental.
Okay, I think we have time for about one more question. I know there's a lot more questions, and we will compile all the questions and send out the answers. But the final question, if SFB mice are housed with non-SFB, which microbiota will be expected to dominate over time, and what length of time is required for cross-colonization when you co-house?
Yes, so cross-colonization is easy as long as the mice come in contact, but it only occurs directly from mouse to mouse. In other words, we have kept cages with SFB, without SFB, on the same rack, even side by side in a ventilated rack. We never see if they're handled properly, we never see cross-contamination or cross-colonization. However, if mice are put together in the same cage, the colonization with SFB happens within a few days. We have done the kinetics for that. If you place SFB positive mice and SFB negative mice in the same cage, the SFB negative mice will acquire SFB within a week. They can be detected within 4 days, but they will be fully colonized to almost normal levels within a week. So I think that's in general true will be true for other bacteria. But when mice are co-housed, their microbiota will generally equilibrate somewhat.
Okay, great. We're going to wrap up. Thank you, Dr. Ivanov. I will be compiling the questions and sending the information out to all attendees, as well as the recording by the end of this week. Thank you all for attending.